every patient with metastatic colorectal cancer that can't be cured by surgery of liver metastasis, resection, etc., still represents an unmet need. Even though we have a lot of agents available, patients will eventually succumb to their disease, so we need to do better. We need to find a way to improve outcomes for patients. That's clearly our goal. And we need to utilize all the knowledge that we've been generating over the last years in terms of tumor biology, interaction with immunotherapy, and really funnel that into the design of clinical trials. So we need to look at relevant preclinical models that give us signals where we can actually move drug combinations forward into clinical practice, into clinical trials. Clearly, one of the emphasis right now is trying to make immunotherapy work in patients where immunotherapy didn't work before. IO combinations, either for immunotherapy agents that get combined or modulators of immune responses. That's one area. Then secondly, also looking at the molecular profiling of patients, teasing out subgroups of colorectal cancer patients that could respond to targeted agents. There's a BRCA mutation group in colorectal cancer that could potentially be responsive to PARP inhibitors. Clearly, HER2 overexpressing tumors, very interesting data with HER2 targeted agents. The patient BRAF mutations, which represent a natural target. There are interesting studies now with agents that can target certain KRAS mutations, the G12C patients, which make a certain subgroup of patients with metastatic colorectal cancer. So we have specific inhibitors here that can target this patient population. So we are looking at molecular profiling and making immunotherapy work in patients where it didn't work before. I think these are the most exciting areas of drug development right now.
Regorafenib as itself is an important modulator of the angiogenesis and the interaction between stroma and cancer cells, and its multi-kinase activity by blocking different kinase receptors in different types of cells can play a major role. Within this possibility, regorafenib can block specific receptors in regulatory immune cells and in the stroma that in some way can help to reactivate the immune system. Given this very strong potential biological rationale, a series of phase 1 studies have been started. Among these, there is a very interesting study that has been presented for the first time at the ASCO annual meeting here in Chicago in 2019, called the REGO-NIVO study. This study that was presented by a group of Japanese colleagues was a phase 1 classical dose escalation study of regorafenib with a dose starting from 80 to 120 to 160 milligrams per day every 3 weeks with 1 week of rest, with the addition of standard dose nivolumab every 2 weeks. And Japanese colleagues did this phase 1 study in MSS colorectal cancer patients and in gastric cancer patients. Both patient populations were heavily pretreated chemorefractory patients. And then they did an extension after defining which was the best tolerated dose for the combination of regorafenib plus nivolumab in both tumor types for a total of 50 patients. The important message from this study is that although these are preliminary findings that should be confirmed in extended phase 2 studies, eventually in randomized phase 3 studies, the bottom line, the most important message is that this combination was really promising in signs of activity in this heavily pretreated population in which immunotherapy, especially for metastatic MSS colorectal cancer, as we said earlier, has no effect at all. In fact, in about 25 patients with metastatic colorectal cancer treated in this phase 1 expanded study with the combination of nivolumab and regorafenib. By the way, the best combination in terms of activity and tolerability is regorafenib at 80 milligrams per day. So this combination gave an interesting response rate of about 33 to 36%. So 1 out of 3 patients in this trial had a major clinical response. And this is quite remarkable because we expect basically 0 responses by nivolumab, about 1 or 2% responses by regorafenib in this set of patients. As I said before, regorafenib is mostly cytostatic rather than cytotoxic, and nivolumab does not work at all in patients with MSS, metastatic colorectal cancer. Therefore, this about 33%, or let's say 1 out of 3 response rate was very and quite remarkable. More interesting, the duration response were quite remarkable. If I recall well, the median progression-free survival was in the range of 6 months. You should expect a range of between 1.5 and 3 months with regorafenib or TAS-102 in this patient population, and basically no activity of nivolumab by itself in this patient population. And also, the expected survival is relatively longer, although data are not mature yet. Therefore, I think that this rego-nivo combination, in which regorafenib is using to immunomodulate the response to a PD-1 inhibitor at a relatively low dose, 80 milligrams, with a very well tolerated side effect profile, could be a new important option for those patients, MSS, metastatic colorectal cancer, in which there is no hope with immunotherapy so far, because PD-1 and PD-L1 inhibitor by itself has failed. Even modulation with MEK inhibition has failed. So these are good news. Obviously, after this good news, we need a stronger evidence that can be based only on clinically and mechanistically sound further clinical trials. Yeah, what this combination can have as a mechanism of potentiation of PD-1 activity is that regorafenib could inhibit and block some lymphocytes that usually are blocking the immune response. So in this way, regorafenib is restoring a potential immune-competent infiltrate in the tumor, and by blocking PD-1 at the same time, all the cells that could be mounting an immune response to the cancer cells could be fully activated. And so, in at least a subgroup of metastatic colorectal cancer patients whose tumors are considered cold for immunotherapy can be rendered by this combination potentially hot.